Good morning, everybody. Thank you for coming. We're going to get started. We have one school who's coming in. They're a bit late, but shouldn't be more than 5 minutes or so, and they'll quietly be directed up to the balcony, so it won't disturb anything. So I'm Ron Perlman, Dr. Ron Perlman. I'm a professor here at York in the biology department, but I'm also associated with the Gairdner Foundation, who are bringing this program to you today. And many of you probably know very little, if anything, about the Gairdner Foundation. And so I'd just like to take a couple of minutes to say something about the foundation and what they do and why they're bringing this important program and letting you people be a part of it. So they're the group who's bringing this exciting event to you, and as I say, most of you probably know very little about them. So on behalf of Dr. Janet Rossent, who's the president and scientific director of the foundation, I'm actually, my official title, I don't know what it means, but is associate scientific director of the foundation, and Dr. Lauren Terrell, who's the chair of the board of the foundation. It's now in its 59th year, so we'll be celebrating the 60th anniversary next year. It was founded by a gentleman by the name of James Arthur Gairdner in 1959, and he was convinced by friends and family to put his considerable wealth to good use, and what he decided that would be the recognition of excellence in scientific endeavor that leads to good for society. And if you want to have one word that defines the foundation and what it does, it's excellence, the recognition of excellence. The awards are supported by a recent significant endowment by the Government of Canada and now include 3 different awards: the Canada Gairdner International Awards and the Canada Gairdner Whiteman Award, and now entering its 8th year, the John Dirks Canada Gairdner Global Health Award. And your speaker today, who will be introduced formally in a few minutes, is a 2017 one of the awardees, one of the laureates of the Canada Gairdner International Award last year. So these are Canada's most prestigious awards in biomedical science and rank amongst the most prestigious scientific awards in the world. The Gairdner Awards are unique in many ways, and one of the ways they're unique amongst the major awards is the reason you're here today and what they do, and that's a national program. So it brings present and past award winners to speak about their award-winning science at, I think we're now at 22 venues across the country, from Vancouver to St. John's, and at close to 20 of those venues, we speak to groups like yourselves, to high school students. And the program puts scientific excellence at the forefront of Canadian science. So the Gairdner events are a true trans-Canada celebration of scientific excellence. They culminate in Toronto tomorrow evening, and tomorrow actually, with a series of exciting symposia where the award winners speak, all of the laureates speak, and then an award ceremony, a gala black tie award ceremony tomorrow evening where the awards are formally presented. Our laureates do a lot of work for us because they think this national program, and especially working with yourselves, the high school students, are important. Your first speaker this morning did a lot when he was in his laureate year last year, and yesterday he was in Ottawa speaking to high school students in the morning and to faculty and trainees in the afternoon. You'll hear a bit later, our second speaker is a member of one of the peer review panels of the Gairdner Foundation, whose responsibility is to select the excellence and to identify the laureates. The York program has been part of the Gairdner National Program for a number of years now. And our goal in this is to bring to high school students the excitement of this scientific excellence, to yourselves, the next generation of scientists who are the future of scientific excellence. And just to reassure you, there's not going to be a test at the end of this. The idea is not to teach science, but to use the award-winning science to talk more about science in general, about the scientist, how they decided to go into science, who their mentors were, the excitement of doing science, what it means to do science, the importance of building a strong foundation of basic understanding before the applications will come, and the unpredictability of where those applications will come from. And possibly, depending on your questions, providing some advice to students, but mainly to excite you as students about biomedical science and the possibilities of that as a career that you might want to think about. Later this afternoon, we'll have a Gairdner lecture, which is a research presentation to faculty and trainees. Now, many of you may know, and many of you may not know, but the Gairdner Awards are excellent predictors of Nobel Prizes. Nearly 30% of Gairdner Award winners have gone on to receive Nobel Prizes. And recent history speaks to especially strong performance in the Gairdner Nobel continuum. This year, for example, the Nobel Prize in Physiology or Medicine was announced, I guess, almost 3 weeks ago now, and was awarded. One of the two recipients of that prize was a 2013 Canada Gairdner International Award laureate for his work in cancer immunotherapy. And this same year, this year, 2 days after the Physiology or Medicine Prize was awarded, the Nobel Prize in Chemistry included 3 people, one of whom was a 2014 Canada Gairdner International Laureate. So in the 59 years of Gairdner's existence, now 89 Gairdner laureates have gone on to receive Nobel Prizes. So certainly that's wonderful for the laureates, but what it tells us, and I hope you, is that our peer review process in selecting the excellence of science is very strong and very robust, because we precede prizes like Nobel. We can't do these things without sponsors. I guess that slide has been taken off, but I'd like to recognize, as I've mentioned, the Government of Canada, the Government of Ontario, Canadian Institutes of Health Research, Burroughs Wellcome Foundation, the Globe and Mail is the media sponsor, plus the universities that we visit, York University this morning, and their staff who do the logistics and put this on. So in a minute, well, it's my pleasure again to welcome you here today, to thank you for coming, and I know you'll enjoy this exciting event. And now I'd like to call on a student at York University, a fourth-year honors biology student, Ms. Vannie Abraham, who will introduce our speakers. So we'll have one speaker and then a Q&A, and then our second speaker. Vannie.
Hello and good morning, everyone. My name is Vannie Abraham, and I'm a fourth-year biology student here at York University. It is my privilege to welcome you to the 13th Annual Gairdner Awards presentations at York University. We're excited to have you here to gain some insight into the field of research, and I hope you feel inspired by hearing from some of the best the field has to offer. The Gairdner Awards, as you've already heard, are Canada's most prestigious biomedical research awards, recognizing the hard work and dedication demonstrated by researchers worldwide. Research is a field born out of our fundamental desire to understand the world around us. Questioning currently accepted thinking, filling in the gaps in our knowledge, and pushing the boundaries between the known and unknown help to answer basic questions about our world and ultimately advance humanity. My own research opportunity here at York University has been an invaluable learning experience. I met my research supervisor at a Meet Your Professors event in my second year. With a semester of introductory genetics and biochemistry fresh in my mind, I was captivated when listening to professors speaking about their research in topics I had previously only read about in textbooks. After being awarded a York University Dean's Undergraduate Research Award and an NSERC Undergraduate Research Award, I've had the incredible opportunity of spending the past 2 summers working in Dr. Ron Perlman's molecular biology lab here at York, and will be continuing with an honors thesis. My research experience has not only been an experiential learning activity, but has also taught me patience, perseverance, and critical thinking. Many of you are probably in the process of deciding which universities and which programs to apply to, and I remember being in your shoes, feeling a combination of the excitement of having so many choices, as well as being intimidated by the challenge of having to choose the best option for me. You stand at a point in life where anything is possible, and with determination and passion, you have the potential to achieve great things. And I can think of no better way for you to recognize the heights that you can aspire to than being here today, hearing from 2 leading scientists whose dedication to the pursuit of knowledge has established them as revolutionaries in their fields. With that said, it is now my honor to introduce our first speaker, Dr. Louis Kay, who is a laureate of numerous accolades for his research contributions, including the 2017 Canada Gairdner International Award, as well as the 2018 NSERC Hertzberg Gold Medal for Science and Engineering, which is the Natural Sciences and Engineering Research Council of Canada's highest honor. Dr. Kay earned his Bachelor of Science in Biochemistry at the University of Alberta, his PhD in Molecular Biophysics from Yale University, and he later went on to work at the National Institute of Health as a postdoctoral fellow. He is now a professor in the departments of molecular genetics, biochemistry, and chemistry at the University of Toronto, as well as being a senior scientist at Sick Kids Hospital. Dr. Kay is an accomplished scientist who has published over 400 papers to date, and is widely known for his work in nuclear magnetic resonance, or NMR, spectroscopy. The research in his lab focuses on the development of NMR techniques, using powerful magnetic fields to study the structures and functions of macromolecules, such as proteins. These techniques have given us understanding into how the formation of abnormal structures by molecules involved in neurodegeneration can ultimately lead to disease states. His research has also extended our understanding of how cellular machines function and has clinical implications for the development of drugs that can target the communication between different parts of these machines to aid in the fight against cancer. Moreover, the groundbreaking methods developed by Dr. Kay are freely available to be used in labs around the world, facilitating the advancement of many other realms of research. Dr. Kay is here today to speak to you about his work in a presentation entitled Watching Proteins Dance Using Biophysics. Please join me in welcoming Dr. Louis Kay.
Well, thank you very much for that lovely introduction. I'd like to begin today by asking you guys a few questions, if that's okay. And you can show me, I can barely see, but you can show me by raising your hand if the answer is yes. How many of you are fascinated by math and science? Excellent. How many of you find that high school science is a bit too cookbook, and you're wondering if science is at all creative? Some of you. Don't worry, your teachers are not taking note. How many of you would like to become scientists? A few brave souls. Why am I asking so many stupid questions? Well, the answer is that I have 2 kids, a 20 and a 15-year-old, and my 20-year-old recently graduated from high school. And what he tells me is that English is way better than science. He says you can be super creative. The teacher gives you an assignment, gives you a blank page, and you get to fill it up with words on essentially a subject that is of interest to you, and you get to put the whole thing together. By contrast, he said, science isn't very creative. For example, you're given a problem like this: to 20 ml solutions of say water, you add 5 mg of potassium permanganate, you stir vigorously for 15 minutes until your arm falls off, you add 2 mg of compound X, you watch the color change, and then you write a report. Does that seem a little bit familiar to some of you guys? Okay, so what I'm here today is to tell you that science is, in fact, very creative. And if you're curious and hardworking and clever, it might just be for you. Let's come back to writing that essay that my son says is an example of creativity. And it's true. You got a blank page, you get to be creative, but think about it. You've been training for 16 or 17 years of your life to write that essay. You speak English all the time, you practice your words, you know them, you were taught them, you went over them, you made mistakes. But in fact, there's a lot of words. I mean, if you look at the authority for the English language, that's the 20-volume Oxford English Dictionary, of which there's over 170,000 different words. And if you put the words in all the different forms, nouns, adjectives, etc., there's about 750,000 words. So before you write that essay and show the world your creativity, you have to have practiced an awful lot for decades, for at least a decade or two. In contrast, there's only 118 elements in chemistry. So it seems like that's a lot easier. Let's take a different example. How many of you are athletes and play football? Okay, just 1 or 2 of you. So let me review the basics of football. So in an average playbook, there may be 30 run plays and a couple hundred pass plays. And there are variations on the theme, so there's well over 1,000 plays. And you've got to be able to essentially react very quickly at full speed, knowing that you're probably going to be hit by some really big person that's 300 pounds that wants to destroy you. Now that's hard work. By contrast, how many elements are there? 118. So you see, chemistry isn't that hard. So I want to just ask one last question. Would you rather read a chemistry textbook, and even if there's a page per element, it's only 118 pages? I mean, that's sort of like bedtime reading. Or would you prefer 20 volumes of the Oxford Dictionary, only 170,000 words for your entertainment? Or would you rather be tackled by a 300-pound Superman? So maybe science isn't that bad after all. Whatever you do, whether it's football or other sports or science, or law or what have you, being a teacher, it requires a lot of training and hard work. And science is certainly no different. You've got to learn the rules of the game before you can effectively play the game. So what is it that I do, and why should anybody care? So my research is at the crossroads of 2 different fields that sort of fertilize each other. One is physics, and the other is biochemistry. And I try to invent ways of solving hard problems and hopefully interesting problems that are of interest to me. And I try to be as quantitative as possible. So these are interesting problems in biochemistry that are solved via the tools of physics. Now, I have a confession to make. When I was your age, I used to put a math book under my desk in English class. I'm sure none of you would ever do this. And while the English teacher was talking about Shakespeare, what have you, I was doing math problems. And I thought I was being very clever until I had to take an exam to get entrance into graduate school in English, and I only got 65%. So I want to make it very clear that whilst it's great to be really keen in science, you should be keen in all sorts of different subjects, because English is just as important. You should have a curiosity really for the world, of which, of course, science is a big part. So what I'd like to do now is educate you a little bit about what it is that I do and what it is that I find fascinating. And to do that, I have to take a step backwards and tell you a little bit about biology. Now, are most of you taking biology, or you've taken biology? Can I have a show of hands? Okay, a large number of you. Great. So then you'll know that inside a cell, there's going to be a nucleus, and in the nucleus is going to be DNA, which is the blueprint of life. And the DNA is going to code for proteins, and proteins are molecules in the cell that carry out the function. They're the movers and shakers of cellular activity. And what I study are these beautiful protein molecules, what their shapes are, how their shapes change with time as protein molecules interact with other molecules. Now, protein molecules are really important, and that's illustrated here on the right-hand side of this slide. Proteins are essentially, antibodies are made up of proteins, and antibodies are very important, of course, for ensuring that we have good health, for essentially attacking foreign objects in the body, antigens. Proteins are also cellular messengers. They're involved in transmitting a signal, for example, across a membrane, and then downstream from that signaling process in the cell. Muscular proteins are very important, proteins like actin and myosin. Proteins are involved in the composition of hair and nails. Proteins in the blood are responsible for the transmission of oxygen to other organs in your body. Proteins are important components of the brain and nerves. Again, you have ion channels transmitting information. Proteins ensure that the reactions of life can occur in a timely manner. Many of the reactions in the absence of proteins would take literally hours or days to be happening, and enzymes allow these reactions to occur very rapidly. And proteins can also serve as construction workers. They build up the various machines that are necessary for activities in the cell, and then they build them down after their activities are done. So proteins do much of the work in the cell, and again, DNA encodes for ultimately these proteins, and these proteins then carry out function. So given the importance of proteins, I thought we would spend a little bit of time looking at them in detail. Have some of you studied about proteins in your classes? A few of you. So let's just do a quick review. Think about beads on a string. And suppose that we can have 20 different beads. So these beads are going to be coming in different flavors. There's 20 different flavors of them. And these beads are essentially amino acids, which are the basic constituents of a protein. And these amino acids are made up of carbon and nitrogen and oxygen. So every circle here corresponds to a bead, and these beads are going to attract one another or repel one another, so that for the majority of proteins, the attractions and repulsions gives rise to a three-dimensional structure. And that structure is absolutely critical for function. Some proteins, we now know, actually don't assume a structure, but are sort of moving in and out between a variety of different structures. They're sort of like a bowl of spaghetti where the spaghetti is wiggling and waggling, or a snake, if you like, that sort of slithers around. And there's not a defined structure, but in fact, these proteins are very important in signaling and in cancer. So the majority of proteins, however, do have a structure, a set of structures. The structures change as a function of time, but it's the interactions between these building blocks that gives rise to those structures. And I want to just show you a few of the beautiful structures that proteins can assume. So this is a protein called calmodulin, and calmodulin interacts with other proteins. Proteins interact with proteins and with other molecules to generate a response. Calmodulin can interact with metal ions, shown here are a pair of metal ions at the top and the bottom, and it can also interact with other protein molecules. For example, here is calmodulin without any other protein, and here is calmodulin with a red segment that is bound to it. And you can see that there's a big change in the structure of the protein upon binding its target. So proteins can be manipulated. They're very malleable in response to interactions. Here's an antibody molecule, and you can see that you have like two balloons, and this is the base, and you have very flexible regions here. And so these regions of the protein are going to move about, and that's very important for the structure. So proteins are not static entities, but they're moving about. Proteins are also involved in the capacity of molecular machines. They're associated with steering cells towards food, for example, or away from danger. They're also involved in printing money. And one of the currencies in the cell is a molecule called ATP, which contains energy and is the energy source for providing cellular activity. This protein is involved in generating ATP. Proteins, in combination with other biological molecules, are often involved in deciphering instructions, for example, for making additional proteins. And they can also serve as gates, ion channels, that allow the transmission of the signals across biological membranes. Proteins can also serve as garbage cans. essentially degrading other proteins once they've carried out their function. This is really important. If a protein finishes its function and then just stays around in the cell, and if it gets diseased and sort of opens up a little bit, and you have sticky regions of proteins that interact with other sticky regions of the protein, that can give rise to fibers and various diseases that are associated with them. And this giant molecular machine called the proteasome is going to chop up those sorts of proteins. Proteins come in different sizes, so this is the first molecule that I described, a molecule called calmodulin, which is very small compared to this huge garbage can that exists in cells. Proteins are involved in reading and delivering mail. For example, there are receptors. These receptor molecules can interact with other proteins, which essentially give rise to a signal. The receptor molecules can interact one with the other, and that can cause some sort of downstream signal. And in this way, you can transmit information, again, across a biological membrane. So proteins are not static, and we can't capture their function by just looking at a single picture. So what I try to do is to understand how the structures of protein molecules change as a function of time. You might say, why does this matter? And so let me illustrate that to you. I show here a picture of a couple of babies. Suppose that you come from a different planet, and on this planet there are no babies. Then you're brought to Earth, and this is the picture that you see of a baby. You've never seen a baby before. And so you can draw some conclusions. All babies are happy. Babies can feed themselves, and babies always wear clothes that are green. And to some extent that's true, but there's a lot of falsities associated with those statements. So you go a little bit further. You find out that babies, in fact, like spaghetti, and moreover, they like to play with spaghetti, but they look pretty happy. And then I show you another picture, and you realize that babies are not always happy, and they're not always wearing green. And so the bottom line is, if we want to understand babies, we have to look at babies as a function of time. We can't look at any one static picture, but we have to take the ensemble of pictures, if you like, to really allow us to understand what it is that make babies work. And the same thing is really true about molecules. Multiple states, multiple pictures are needed to understand a baby, and the same situation occurs in proteins. So what I show here in red is going to be an enzyme, and in blue is a molecule upon which the enzyme acts. It's called a substrate molecule. A chemical reaction occurs, and then you get breakage of this blue substrate molecule. But the point is that the protein, the enzyme, has had to change its shape, as you can see, to accommodate the substrate molecule. Now, proteins don't need an excuse to be dynamic. They're dynamic by themselves. So let me just illustrate that in this movie as well. So remember that proteins are made up of amino acids. These are these beads on a string. Each amino acid is in turn made up of various atoms, hydrogens, carbons, and nitrogens. And these amino acids are arranged into structural features like helices and like strands, and you're about to see them in just a minute. Here are these beautiful yellow sheets. Each one is a strand of a sheet, and you can see the purple helices. And although this protein is depicted right now as being in isolation, in a cell you have a very dense accumulation of a whole bunch of different molecules, including different protein molecules, including water molecules, including small substrate molecules. And the protein is going to be bombarded by all of these molecules. So imagine that you want to go from point A to point B, and there's a bunch of people in between. You're going to bang into these people. And as you bang into these people, there's going to be an exchange of energy between you and all of them. The same thing happens in the protein molecules. And this energy is going to converge in a particular way, which results in function. So in order to understand how these protein molecules function and misfunction, we need to understand how energy is going to be transmitted from one molecule to the next molecule and guided along a particular path to carry out a particular reaction or a particular function. So what I try to do is understand how molecules dance. And I use magnetism to do that. Are you all taking physics? Some of you must be taking physics, a few of you. Can you show me by hands? So there's a few brave souls out there. So let me just review a little bit about magnetism and tell you why I've decided to choose magnetism and magnetic fields to understand the beautiful dance of protein molecules. It seems sort of strange, you might think. And I got my inspiration from this guy, Dave Barry, who is a world-famous physicist. And this is what he says. Magnetism is one of the 6 fundamental forces of the universe. So you can see that magnetism is really important. The other 5 being gravity. Well, we all know about gravity. Duct tape, whining, remote control, and the force that pulls dogs towards the groins of strangers. But I note that magnetism is number 1. So you see, if you want to do something, magnetism is a good way of doing it. Let me just review some of the basic features of magnetism, which we all know, but are going to be important for describing what it is that I do. Well, we know that magnets have both a north and a south pole. And we know that there's an attraction between north and south poles, and there's going to be a repulsion between a pair of north or a pair of south poles. That we know. Now, what I do is as follows. I spend a lot of money, costs over $1 million, to buy a giant magnetic field. Now, the Earth gives off a magnetic field as well, but my magnetic field that I spend millions of taxpayers' money for is about 200,000 or 300,000 or 400,000 times the Earth's magnetic field. So I have a giant magnetic field, and like other magnets, there's a north pole and a south pole. Then what I do is I take these protein molecules, which are dissolved in solution. Now remember, these protein molecules are made up of atoms, and it turns out that every atom in the protein molecule is essentially a bar magnet. So it has a north pole and a south pole as well. Now the physics to describe these little tiny microscopic bar magnets that are embedded in every protein that I study, because they're associated with the nuclei of the atoms, and there's a lot of atoms in the proteins that I study. Every one of these bar magnets obeys the laws of physics. I need quantum mechanics to understand the little bar magnets because they're microscopic. But essentially, it's very similar to the sorts of things that you would be familiar with the macroscopic bar magnets that you may attach to your fridge. So let's look at the following situation. What I have is a solution of atoms. Each atom is a little bar magnet. And what's going to happen when I take my solution and I put it in this big magnetic field? Well, the bar magnets are going to orient. So the south poles of every one of these little bar magnets that are associated with the nuclei of the atoms in the protein is going to, they're going to align with the north pole of the big bar magnet. The majority of them will, and a few of them will anti-align. That is north with north and south with south. It turns out that quantum mechanics tells us that there's only 2 orientations of the bar magnets, up or down when you put them in a big magnetic field. Most of them are going to be in the lowest energy position, so that's north pole with south pole. And then what I'm going to do is as follows. Most of you are familiar with punching bags, right? Some of you may even have punching bags. If you have a bad day at school, you don't want to take it out on your parents. After all, your parents are good people, but you come home and you have a punching bag and you punch the punching bag. So let's assume that our little microscopic bar magnets, our little nuclei, are much like punching bags. And what we're going to do, just like when you come home and you punch the punching bag initially before you punch it is at its lowest energy state, its equilibrium state, it's upright. Then you put energy into the system and the punching bag goes down, and then it comes back up. That's what we're going to do. We're going to put energy, magnetic energy, into our spins. They're so-called spins. These are these little microscopic bar magnets. They're like punching bags, and we're going to punch them over. And then we're going to watch them as they come back to the equilibrium position, which is this position here. And it turns out that these little bar magnets are going to talk to one another. We know that if we bring a north pole close to a south pole of 2 bar magnets, there's an attraction. But there's hardly attraction if the 2 poles are very far away. They have to get close for an attraction to occur. Same thing here. The interactions between these bar magnets depends on distance. We can study the interactions by watching how the bar magnets come back after we've punched them, and therefore we can learn about the distances between the bar magnets or the distances between the nuclei of the atoms in the protein, and therefore get structural information. And moreover, we can get information about how the structures change as a function of time. So just to emphasize that again, we have a north pole and a south pole of our very big magnet that's a few 100,000 times that of the Earth's magnetic field. We have a sample that consists of 10 to the 17th little bar magnets. That's a huge number of little bar magnets. Most of them have the South Pole associated with the North Pole and the North Pole associated with the South Pole of the big bar magnet, and then we apply a punch. We can do all sorts of different punches. We can apply a punch to the left, a punch to the right. Again, think about the analogy with the punching bag. We can punch it on the front end, the back end, on the sides, any position that we want. We can apply a big punch or a little punch. We can apply all sorts of energies into our molecules for long periods of time, short periods of time, to essentially learn information, tease out information about the molecules that we've studied. Let me show you how it works. So here I show an NMR sample, and you can see at the base of the sample we have a solution. This solution contains our protein molecules whose dance we want to study. And like I said, there's something like 10 to the 17th of these molecules. This is what a very big magnetic field looks like, and we take our sample and we put it inside the magnetic field. Over here we have a lot of electronics which essentially provide the punches to the little bar magnets that are associated with the nuclei of the molecules. And remember, we have a whole bunch of different molecules, and we're then able to learn something about these molecules. We record what is called spectra. Each one of these dots in this two-dimensional representation is from an amino acid, so from one of the beads on the string. And we can analyze patterns like this to get out the distance information between these bar magnets, because remember I said that they interacted, and like two magnets interact in a manner that depends on the distance between these bar magnets, so too we can have interactions between these microscopic bar magnets that allow us to measure distances and hence to get out three-dimensional structures. And over many years, our laboratory, as well as others, have developed experiments that allow us to get really very crisp pictures of what these biological molecules might look like and how these pictures change with time. But there was a dark cloud that emerged on my research. The problem is that as we go to bigger and bigger molecules, and I want to study the molecular machines in the cell, and these are really very big molecules, the signal decreases very quickly. So imagine, again, the analogy with the punching bag. Suppose that you punch the punching bag and the punching bag goes down, but then it comes up so quickly that you can't detect that it was ever down. That's what happens as we use this NMR technique to study the structure of these very, very big molecules. We simply lose the signal so quickly that there's no information whatsoever. And so what we had to do is we had to think about designing experiments that would allow us to get the signal back. So we want to study these biological molecules, but we're going to have to use physics to somehow tease out the signal from them because the signal is decaying away too rapidly. So let me just show you examples of different sizes of proteins. So here we have a baby protein that is made up of only 50 or 60 little amino acids. So only 60 beads on this particular string. Here we have a protein that is 10 times bigger, made up of about 800 beads on a string. And here we have a protein system that is a factor of 10 larger yet. Now, we can study these little proteins using traditional experiments. That's not too hard. But if we want to study the real big systems in the cell, we've got to come up with ways that we can actually detect the signal before it decays away so rapidly. And so what we've done is we've come up with physics that, if you like, tricks the little bar magnets, the microscopic bar magnets that are giving us the information about the molecules that we want to study. We trick these little bar magnets into thinking that they're actually associated with a small molecule, a small protein, rather than a big protein. So we can use physics ideas to trick these little bar magnets into thinking that they're part of a small protein rather than a big protein. And that allows us to restore the signal that would otherwise be lost. And when we do that, we can begin to study very big protein molecules. And I want to end my presentation by just showing you one such protein molecule that we study. It's a protein molecule called P97. This is my rendition of a cell. And I want to focus your attention on this molecule, P97. It's a very important molecule because what it does is it takes other proteins when they're past their prime or when they're damaged, to the cellular garbage can that I showed you a picture of before called the proteasome. So remember, when proteins have done their tasks or when they're diseased, you've got to get rid of them before they can do damage to the cell. And P97 is a molecule which picks up that damaged protein and transfers it to the garbage can. How does P97 actually do that? How does it pick up the diseased protein? Well, P97 has arms, just like you and I do. P97 has 6 arms. And you can imagine that the position of the arms is really important for being able to interact with other molecules that are diseased and transfer them to the proteasome to be degraded in the same way that you use your arms to give a person a hug or what have you. P97 has to use its arms in a particular way. Now, it turns out that there are mutations in this molecule, in this red P97 molecule, which can cause very significant neurodegenerative diseases. The P97 molecule is made up of a set of strings. Each string has 800 amino acids, so 800 beads, and there are 6 copies of that string that make this molecule. And you can have just a single mutation, 1 out of 800 being wrong, and that causes very severe diseases. So if you take an exam, for example, and you get 99 of 100 questions right, you go home and you tell your parents, look, I got 99 out of 100 right. What are they going to say to you? What happened to that last one? No, they're going to say, great job. Now imagine that you get 799 of those questions right. I mean, that's amazing. You guys are geniuses. So what happens with P97 when 799 amino acids are right and only 1 is wrong? What's your reward there? You get a very serious neurological disease that causes death. And the reason that it causes death, we were able to figure out, is because of the arms. Just like the arms are important for doing things like your arms are, what happens is that these particular mutations, and you only need 1 of them, essentially distorts the function of the arms. When the arms are supposed to be down, they're up. When the arms are supposed to be up, they're down. And so the arms don't interact with other molecules in a proper way. We were able to figure that out with the tools of biophysics. And then what we were able to do is take another amino acid, one additional one of the 800 amino acids, and change that one, and in so doing, shift the arms back to their wild type or non-disease state. So we took a very diseased protein, made one little tiny change, and shifted the function of the protein back to the non-disease state. And so this gives us confidence in terms of a proof of principle that maybe we can develop a drug which will just touch the protein, this diseased protein, in a specific way, and will push the arms back into functioning properly, getting rid of disease. So this is an example of how one can use physics to understand a biological problem. So let me conclude by saying this. Science is creative. Learning the rules is really hard, and I feel for you guys. You guys are now in the stage where you're learning the rules, but once you know the rules, you'll be able to play the game, and that's a lot of fun. Don't get discouraged. In chemistry, for example, there's only 118 elements, which is a lot less than 750,000 words in the dictionary. Choose your passion. Any passion is equally good. Listen to your teachers, but not too much. I remember when I was at your stage and we were doing acids and bases in chemistry. Do they still teach you guys acids and bases in chemistry? So the teacher went to the blackboard and he wrote down, this is the way you solve an acid and base problem. You know, step number 1, you do this. Step number 2, step number 3, and so forth. And at the end of the lesson, I went up to the teacher and I said, you don't actually expect me to follow your rules to solve acid and base problems, do you? And I think the important thing is solve problems your way. Each and every one of you have a unique way of solving problems. It's different than anybody else's ways of solving problems. And that will allow you to answer questions that really nobody else can. And that's really critical. And of course, you can do the work. Don't let anybody tell you that you can't, because you most certainly can. And good luck to you. Thank you.
Okay, thank you very much for that tour. We do have time for a few questions. There are two people at the back with hand microphones, so please use those. Anybody in the balcony, could you please come down to access the microphone? So as you've seen, it's hard to see here, but raise your hand and we'll get somebody.
Well, I know that people are a little scared and nobody wants to ask a stupid question, but there's no such thing as a stupid question. I used to ask a lot of questions when I was your age. I still ask questions. That's what we do for a living. But let me ask you one question. I asked the question initially, and that was how many of you wanted to be scientists, and some of you put up your hands. How many of you now want to be scientists? Okay, so one. Okay.
Hello. Thank you. So my question is, you're using MMR to generate that magnetic field, which is obviously very intense. But is there magnetism within cells that drives these protein shape changes? And if so, what generates it?
Right. So these shape changes are caused by forces. There's no doubt about that. There's a lot of different forces that exist in a given protein, and they're essentially all add up to give rise to a structure. There's forces between positive and negative charges, electrostatic forces, for example, because some of these beads on the string are positively charged, some are negatively charged. There's also interactions that exist that are electrostatic in nature that are not caused by charges that are stably formed, but charges that are transiently formed, van der Waals interactions as well. But there are also interactions between different molecules which cause a distortion in the shape. So for example, if you're walking and somebody bumps into you, right, and your arm swings back, that's energy that he gives to you. Now that happens in the cell all the time because the cell is a very crowded environment. There's literally hundreds of milligrams of protein material per ml in the cell. So there's a lot of proteins in the cell, and these proteins are bashing into each other. And not only that, there's a lot of small molecules in the cell as well, and these small molecules are bashing into proteins. So there's a lot of exchange of energy which causes the protein molecules to get distorted in a fairly stochastic manner, in a manner by chance. But it turns out that that allows the protein to explore different areas of its conformational space, or to explore different shapes and different forms. And it may be that those shapes or forms are going to be those which are responsible both for function and misfunction. Let me give you an example. Suppose that you have a protein molecule. Now the protein molecule, most of them are stuck together through interactions, but over time, maybe the interactions become a little bit weakened through mutations or through loss of various cofactors like metal ions that generally would bind to the protein. So suppose that I'm the protein and I have an interaction between my arm and my chest. Over time, the arm opens up, and that exposes sticky regions, which can then interact with other sticky regions in a stochastic process, but that may seed the development of a fiber which could cause some sort of neurodegenerative diseases. So there are a lot of stochastic interactions that can occur which can change the shape of molecules. These interactions can be mediated through mutations or through loss of metal ions or through age, as proteins age. And in our body, we have chaperones which prevent that, but of course our chaperones are proteins too, and they get old as a function of time, and they decrease in numbers as a function of time, which exacerbates the problem.
Okay, do we have another question or two? We've got a bit of time left.
How did you disturb certain amino acids and not other ones when you sort of say you punched them? So how were the other orientations not disturbed while you tried to affect certain ones with the punch?
Okay, so there's a number of different ways we can do experiments. I can apply a really hard punch, like a crushing body check in hockey, right? Your whole body feels that. So I can apply a very strong punch where every magnet in the protein feels that punch. And I've got a lot of different magnets, but then I can analyze so I can get information about each of the magnets independently. I can do higher dimensional experiments. What I showed you here was a two-dimensional experiment. Do you remember the sort of two-dimensional thing with dots on them? I can turn that into a three-dimensional image or a four-dimensional image, and I can therefore disperse the dots. So you can imagine if you're in a two-dimensional regime and you've got a lot of dots, there's only a finite amount of space. But if I go to a three-dimensional regime, now I have more space, and in 4 dimensions, and 5 and 6, yet more. So I can disperse these dots and apply a strong punch. But I can also apply weaker punches. I control the strength of the magnetic field that I apply to my punching bag, if you like, the strength of my punch. And in so doing that, I can control what amino acids are going to be affected by my punch. So I can do all sorts of things. I can control if only a few amino acids see the punch or all of them.
So when you get the video of your protein dancing, what you're getting is basically all the different kinds of orientations the proteins can take based on what you do to it. And those are the possible movements that can happen in a cell. And so you're looking at the, over time, what happens to it in terms of aging or perhaps interaction with other organelles or other molecules in the cell. Is it all about over time and trying to prevent that, or it could be mutations that can cause these changes to happen in the protein?
So it's all about understanding how the dynamics is responsible for its function, both in a healthy system and in a not so healthy system. Now I should say that, well, I've given you the analogy of a punching bag that really, and a punch that really changes how a punching bag looks. The actual energy that I put in when I apply these really weak magnetic fields is not going to be strong enough to change the shape of the protein, but allows me to look at how the protein would normally change its shape as it evolves, doing its own thing. So I don't really perturb the system, I merely look at it to try to understand how it is that the system is evolving. Think about an MRI. MRI is the cousin of NMR. It's just they've taken away the nuclear part because it makes patients very scary when you think about having something to do with nuclear. But in an MRI, of course, it's fairly non-invasive. We're looking at a tissue as opposed to a molecule, but it's the same sort of physics. We simply tickle the tissue, if you like, to be able to see it without doing anything really invasive to the tissue. And the same thing with NMR. I look at my molecules, I see them dance in their natural kind of state, but I don't punch them to make them dance in a given way. I explore their own dance through various experiments that I do.
Okay, I think we actually better move on because time is a little bit tight at the back end. So we'll thank Dr. K again. And I'll ask Ivani to come back up and introduce our second speaker.
I am now pleased to introduce to you our next speaker, a true clinician scientist, Dr. Nada Jabado. Dr. Jabado is a staff physician in the Division of Hematology and Oncology at the Montreal Children's Hospital, a professor of pediatrics and human genetics at McGill University, and a principal investigator at the Research Institute of the McGill University Health Center. She completed her medical degree and residency in pediatrics with a specialization in hemato-oncology in Paris, and further obtained a PhD in immunology, following that with a postdoctoral fellowship in biochemistry at McGill. Dr. Jabado has earned numerous awards recognizing her contributions to medical research, including the Canadian Cancer Society William E. Rolls Award, and being inducted a Fellow of the Royal Society of Canada. Dr. Jabado's research is focused on pediatric brain tumors and aims to better understand the genetic and epigenetic signatures of pediatric astrocytomas, a deadly group of brain tumors. Revolutionizing her field, Dr. Jabado's lab was among the first to identify a histone mutation as an indicator in human disease. Epigenetic markers such as these, which are heritable changes in the gene function without changes in the DNA sequence, were a previously unknown hallmark of cancer development. This discovery has led to what we now refer to as epigenetic-driven cancers. Her research accomplishments have established her as an international leader in the field of pediatric brain cancers, and she aims to apply these findings to optimal therapeutic targeting of a tumor by its genetic signature, ultimately improving detection and treatment of these tumors. Dr. Jabado has joined us here today to speak to you about her work in a presentation entitled Oncohistones, How to Turn a Cell Symphony into Non-Harmonic Rap. Please join me in welcoming Dr. Neda Jabado.
Good morning, everyone. This is a first for me. I'm going to speak without any slides. So you're going to bear with me and you're going to need to listen to me, because if you are looking for anything to kind of distract you from what I'm going to say, tough luck. So I'll start first by thanking you for being here. How many volunteers to be here? Raise your hands. Obviously, you all are here because you're kind of forced to by some program. And when I was in your shoes, that was a good day out of school, out of, just listening to a teacher speaking about amino acids, about physics, about an essay in chemistry or an essay in English. What I would urge you to do, and again, this is a suggestion, take it as an amazing opportunity to see what could be outside of your own zone of comfort. You're at a time where, you have a lot of doubts. I don't want to be again in your shoes. Even though I want to be younger, every day as I'm growing older, I want to be younger. I don't want to be in your shoes because it's a time where there's so much doubt, so many things, the marks, so much pressure to have, to achieve things, to go, to grow. Can I do it? Can I not do it? Why am I not doing so fine? The pressure from your parents, your pressure from your fears, the pressure for yourselves. But at the same time, it's an amazing moment to live. Mark my words for that. A lot of things are open. A lot of things are possible and can happen. It takes a little bit of perseverance. You don't need to be the super smart. As long as you find a passion, a way, something that really you want to do, and you go into doing it, this is what you should be doing. And you're here to hear about science, and maybe this is something that you could contemplate. Even if you don't want to take it as part of the future you want to embark on, maybe this is something that you should foster. And my role here is to tell you why science is something that's very important to us, even if we go and become businessmen, agriculturists, whatever we become. Because this is part of us. This is something that we will need at every given time point, for us and for our own. Okay, I am a physician scientist. You've heard about all of the things. I didn't start this way. I have 3 kids. They are age 19 soon, 18, 17, and 16. There are some of them at the same stage that you are. They're looking at me and they're saying they want to become doctors. No, it's not because my husband and me both of us are doctors, not because we like what we do. That's the best way for you to do it. I'm Lebanese. My first name doesn't imply, if people think I'm Spanish, and my parents were crazy to call me Nada, like nothing. But I'm Lebanese. I was born in Lebanon at the time of war, and my parents very fast left, and I grew up in France. Lucky. So I'm an immigrant. I potentially could be put as a refugee. Well, even if with that, you could kind of make your own way. I was lucky to have people that supported me, my parents that supported me, and I went into medical school because this is what I wanted. Ever since I knew, I was asked what I wanted to do, I knew what I wanted to do. For some reason, I wanted to become a medical doctor. And this is what I really put every effort into becoming a doctor. I wanted to be a surgeon. I did a lot of general surgery. But then I found somebody who told me that I would be bored, and that maybe doing a little bit of science on the side might be enlightening for me. And I took that advice and I did a master's in biochemistry. which led me to do, on top of my residency, a PhD in immunology, and then to pursue post-career, a clinician career with everything that it entails, and a scientific career with a PhD in immunology, and then a postdoctoral fellowship in biochemistry. And what I learned is to have tools to kind of grow in different environments. So I left France because my husband is Canadian, and I came here, and I came to McGill, because, you know, it's, you go with where the possibilities are, and my personal life was more in Montreal, this is where I live, than it was in Paris, France, and I love Paris, France. You could, why am I giving you all of those things? Just to show you that if you have the will, if you have the tools and the perseverance, wherever you are, you could make it. You start believing in yourself, and you go and you work, and things will happen. If there are obstacles, and there will be obstacles, some you could overcome, but if you don't try, you will never know. So my first advice, if you need any advice, is to try and to acquire the tools to even try better if you fail the first time. If something matters to you, you make it happen. And mark my words, it will happen if it matters enough. So you go, and I am now working on pediatric brain tumors. I never thought I would do that. But then, I'm an oncologist, and when I started, those were the tumors where nothing was known. And this is where I'll speak a little bit of science. So how many of you want to go into the medical world? Raise your hands. Wow, a lot of things. Doctors are overrated. This is very true. There is a huge spectrum of people that take care of people. Your teacher take care of you. The moment you care for other human beings, it's an important moment. It's really a huge responsibility. Those teachers that you have, that you need to listen to, whatever, they care about you because they are shaping your future. They're listening to your doubts. They're trying to put it, and they didn't pay me to say that. This is the reality of things. In the medical field, you have doctors, you have nurses, you have social workers, you have a lot of people that make the care holistic and complete. So don't stop at the term doctor unless really this is where you want to go, because at every step there are multiple different career choices that are equally fascinating and equally rewarding. And this is something, don't go for a title, go for what you want to do and where you feel you can blossom in. And there are many choices, okay? Number 2, how many of you have had somebody in their family or somebody they know with cancer? Raise your hands. Almost everyone. It's a fact of life. After the age of 70, 1 of 2 people will have a cancer. How many of you think that cancer is something that we can prevent? Not a lot. Well, I think cancer is something that's, the most common cancers are something that we can prevent. What is the most common cancer? I'm sorry? Lung cancer. Lung cancer, what is the major cause of lung cancer? Smoke. How many of you smoke? Raise your hand and be honest. Be honest. I'm pretty sure there are smokers over here. Okay, I'll let you be, but again, so don't smoke, and the chances or the risk of getting a lung cancer becomes so low that it's a question of bad luck. So a lot of the cancer, colorectal cancer, breast cancer, and lung cancer, this is really 90% of all cancers, can be prevented. And their incidence from 100% reduced probably to barely 10-15%. The 10-15% that what you're born with, meaning your genetic and the other halves of your environment that you cannot avoid, will have you undergo. If you look in China, what's their first cause of cancer? Or in Asia, mainly China. Lung cancer, why? They smoke, and the cold, the smog. All of this, the particles, smoke that they inhale. So again, cancer is something that is very preventable. How many of you think that we've made headway in treating cancer? Raise your hands. Good? Not too, not. Well, we've made amazing progress. For lung cancer, just based on how, if we catch it early and how we put it and how we now analyze, we're making more and more and more dense. We're actually now seeing side effects, meaning other sites affected by this cancer because people are living longer. The breast cancer is the same thing. Colon cancer, the same thing. We're treating better and better, but the best treatment for now for those cancers is prevention. So I urge you to do that. I work on the 10% of cancer that cannot be prevented, which are the cancers that occur in children and young adults. Because this is something, not that they're born with, but something that they acquire as a fact that, like Darwin said, we are evolving. We are actually tinkering with our genes, with our genome, because each time we try and find another fit, a better fit. Who knows who was the worst enemy we had until about 50 years ago? What was humankind's most unwanted enemy? For example, in Africa, what do they die of? AIDS, infection, tuberculosis, malaria. So infections. We really actually are made of something, it's a struggle against infection. Nowadays, infections are being taken care of. Women can bear children and have safe, at least in modern countries. So this is why we're seeing more and more of these cancers. But the rate of cancer in kids is still the same. This is something that always existed and unfortunately will always exist. And for now, we don't have means to prevent it. The only way we have is to try and detect it early enough and to treat it early enough. The cancers that I treat in children affect the brain, and they're called brain tumors. Within brain tumors, it's like you're speaking about vegetables. There are multiple different vegetables. The one that I treat is the gliomas, the high-grade gliomas. And those, for the high-grade, remain a death sentence within 1.5 years of the diagnosis. So what is it that we've uncovered? Again, as a doctor, in order to treat, I need to understand. To understand, I need to search. And this is why research is at the core of everything. This is why actually I embarked on a research program, not because I love the NMRs, I love tutorials protein, I love physics, I'm curious. At the same time, curiosity is something that is a little bit selfish. But that if I want to help my patients, if I want to help the children and their families, I need to understand what they have. And I need not to make them guinea pigs. So for those patients, for those children, when I started in 2003, my lab, they were treated like adults because they had the same name. The tumor bore the same name, glioblastoma. This is the name it had. It was the same. If you look at it under a microscope, an adult tumor and a pediatric tumor is the same, no differences. So every treatment that was used, and it's not very effective in adults as well, was used in kids and didn't work. So you could continue, try different recipes, try different ones, or you could go back and say, okay, what am I missing here? What should I be understanding better? And the beauty is when you are at a time where you have the tools, and even better tools as we go, to understand what is happening in the tumor, you could potentially make a little bit of a difference. And I'm not the only one, I collaborated. So what I did is I created a consortium that I called iChange, because I love change. I change countries, I've changed from becoming a surgeon, I became an immunologist, and now I'm a brain tumor. So change is something that you should embrace. It builds you. And I had 17 countries. I emailed every single person that worked on those glioblastoma or had treated children with those glioblastoma. And probably I sent about 700 emails. I still have the list somewhere. And asked if they had samples that they could share, because it's a rare tumor and you needed samples, and more samples the better. 7 people answered, send me tumors. And I collaborated with, who is now my professional husband, he's Jacek Majewski at McGill, who had those new machines, the sequencing machines, and we analyzed the tumor and hit gold. By analyzing the tumor and seeing what was really present there, it seems very obvious now, we identified mutation in a gene by the name of histone gene. Anybody have heard of histones? Raise your hands. Okay, not a lot. Anybody heard of DNA? Raise your hands. Okay, great. So Dr. K made it easy for me. So our DNA, do you know how long our DNA is? Anybody can take a guess? 1 meter, 1 meter, raise your hands. 2 meters? 1 here, 3 meters? Okay, it's 2 meter 10. I'm gonna stop it here. 2 meter 10. How a cell is microscopic, the nucleus is even more microscopic. 2 meter 10 of DNA is compacted into nothingness within this. And are you aware that our DNA is exactly the same in every single cell that we have? However, you agree with me that an eye is different from a heart, a muscle is different from a hair, and a fetus is different from an adult. So this DNA that's compacted into nothingness needs also to be expressed, and it's expressed very differently based on the time of life where we are and the type of cells that we have. Because you don't need everything that's in this information to make a muscle. You only need a few genes. You don't need everything to make a given cell within the brain. Only a few of those genes. And this is what those histones help do. Histones form what we call the nucleosome. The nucleosome are protein, you've heard about protein, that are innate, and you wrap your DNA around it. You see like a thread that's wrapped under something, you wrap it. One nucleosome wraps 147 base pair, and all of them are compacted into nothingness. While you're wrapping, you're making this DNA inaccessible except for given area, where it opens and closes to make the gene you need at the time you need it accessible for it's being read and producing the protein that will give you a myosin if you are actin if you're a muscle, or the myelin if you're an oligodendrocyte within the brain and want to have myelin, okay? And this is the beauty of those histones, is they wrap the DNA, but they have a tail that open and close. And what we identified is a mutation within this tail at a residue that's so important that is conserved even in plants, and that were leading to those type of brain tumor. So great, I have this mutation, whatever. It's a new alphabet. It's like the Rosetta Stone. You need to understand what is it that disrupted. And this is why you go back to your drawing board, and this is why research is so key. The way I love about the way we are, this is what I find fascinating. You might find it boring to no end, but for me it's fascinating how from one single cell, remember, we all came from one initial single cell. We have so many million cells across our body and so much diversity. And this is the symphony of our life. This is why those histones actually are, think about DNA as notes of music. Do, re, mi, fa, sol, la, si. Actually, this epigenome, those histones, are making those notes of music be put into so many different partitions. You know how many music there are? There is rap, there is jazz, there is classical music. You have so many different refrains. You could actually, with the same notes of music, make so many different things. And this is exactly what this epigenome is doing with our genetic information, is making it have a given partition, a specific partition for our brain, a specific partition for our heart, a specific partition for the cell when it was one cell and as it's growing. And this is unfortunately was so very wrong in those kids, is that this partition is stuck in time. It's actually, I call it the Peter Pan syndrome. They are stuck in a given time point of differentiation, unable to go forward. They're always young, always multiplying in the same thing, and unable to differentiate, and this is what this mutation is doing to them. So why is that important to understand? It's important to understand if I want to treat. It tells me that if I could potentially push them to differentiate, I could gain something. If I could potentially make this cell that relies on a specific enzyme to keep it in this state, if I remove the enzyme, I could treat it better. It actually gives me new ways of targeting those tumors and gives hope when there was none. Up to now, when those kids are diagnosed with the mutation that I'm telling you about, it's a death sentence within 12 to 18 months. With the finding that we have and other people are having, hopefully within the next 5 years, we can hope for a cure. If it wasn't for research, this wouldn't have happened. And research is so key because you could diagnose things. You could have a heart attack and whatever. Nowadays, heart attacks can be treated. Why? Because we know exactly how the clot forms, how it goes, and we know what to put to go and open the places. This was done through research, by treating patients in a given way, by looking at those effects, by having access to new tools that would allow us to go to the smallest vessel and open it. And we save lives, and we promote a better life this way. And this goes all the time by trying to ask ourselves, how can I do it better? What is it that I'm missing? To go back and ask yourself a question. Is it the right way? Is it always the right way? Can I do it in a different way? Dr. K told you when he was speaking about this equation of acids and bases, everybody will have their own recipe. As long as your recipe is making things better or is making you, this is the recipe you should be applying. And the point here is to always challenge. Don't live on dogma. Challenge and ask yourself, can I do it better? Can I still do it better? And as you learn, you'll get tools to think about it differently. This morning, as I was flying here from Montreal, I was on the phone with my daughter who had an assignment in philosophy. I hate those assignments. The blank page and whatever. I'm like you. It's, for me, words are, I'd rather kind of tackle not a math problem, maybe a physics or a histone problem, but it's the philosophy. So her essay is an opinion capable of being truthful. We all live by our opinion. And again, for us, it's the truth. As long as we challenge it with what we know about it, it's fine. The moment we are set in stone in our ways, I think we regress. And this is what I find beautiful about science, is that you're nowhere in a safe place where that's it, I know everything. The moment you know everything, you need to stop, because that's not true. So many things happen, so many new things are here. I use Drosophila, the fruit flies. You've heard of Sarah Palin? Who heard about Sarah Palin? Sarah Palin was the co-lead for the previous election. She still is, the governor of Alaska, and she ran as vice president in not this election, but the previous one. And what she said about research is that we researchers are crazy people because we use fruit flies and are wasting taxpayers' money on fruit flies. Well, fruit flies are the most economic and most beautiful the most beautiful tool. They're 0-1, they're black and white. My daughters hate me because last summer they spent all of their summer dissecting the eye of Drosophila, the fruit fly. Why? Because you could make this mutation that I told you about expressed in given tissue of this fruit fly and study its effect. And you do it within, the cycle of a fruit fly is barely a week. And nobody cares about flies because everybody tries to kill them. So it's really the best tool ever. Fast, rapid, 0-1, meaning if you have a phenotype, you'll see it. And when we put this mutation in a driver that is expressed in the eye of the fly, the eyes became very small. Why is that important? It allows me, actually, to try and rescue the eye. So what we did, we mated those flies with different flies that has specific mutation here and there to see which one would either make it worse, meaning I want to kill those cells, or make it better, I want to rescue. Remember what I told you, have those cells differentiate, or see what is it that they rely on to stay alive. The eye of the fruit fly allowed me to do so. We have 2 drugs that now we're testing. And this is the beauty of science, is that you could tackle it from many different ways. The other thing that I would like to tell you about, and I'll stop at that, is by being curious, by really, some people told me, okay, you're a clinician scientist, it must be hell because you spend your life in clinic, you spend your life in the research. It's very true that at some point you can't be doing two jobs at the same time. I'm more of a researcher now for the last year than I'm a clinician. However, the foothold that I have in the clinic allows me a perspective that's amazing. Why is that? Because you see patients, you could advise for some of the things, and if there are puzzles, you could potentially take them and help those patients with those puzzles, but also help others. And I'll tell you a story that's completely outside of those brain tumors. I got called about 3 years ago by one of my colleagues who had two siblings, one after the other. The first one came with an immune activation. Their immune system was super active and no nodules under their skin that we call panicolitis. It's inflammation of the fat. And they got biopsy taken, and it was a lymphoma. Lymphoma is the cancer of the lymph node. Very rare, like super rare lymphoma, 1% of all lymphomas. Okay, we treat the kid, he doesn't respond, we give him immunosuppressants, he is in remission, and then because he wasn't responding, the only way is to give him another immune system, and we transplant him with his brother, we give a stem cell transplant, he was actually identical, and he's cured. A year after, his sister comes with the same problem. Brother and sister, genetic disease. Parents were absolutely not blood related. One was from Cambodia, the other one was from Thailand, really nothing in their family history. So we spoke with the parents. This is why my colleagues called me, because I sequenced the genome. I spoke to these families. They agreed on a research basis that we sequenced their genome and the genome of the brothers that was fine, and the brothers that was sick, and the sister that was sick. Within a second, barely, we identified the mutation. You've heard about the checkpoint inhibitors that got the Nobel Prize? There's PD-1, I'm citing one, and the other one is CTLA-4. After PD-1 and CTLA-4, there is another checkpoint that's very, very important. That's called TIM3. TIM3 comes up when you block PD-1, because our immune system, when it's unleashed, we want to stop it. We don't want to have problems with our immunity. So we mount an immune response and we stop it, because after that, if it goes too far, it creates problem to us. So when you stop PD-1 or CTLA-4 or both of them, TIM3 comes up. And actually, in HIV, because the immune system is all the time stimulated, TIM3 is very high. In multiple sclerosis, TIM3 is very high because the immune system is kind of off the mark. So TIM3 is quite important in checking our immunity. The mutation we identified are point mutation, like the protein that Dr. Kay spoke to you about. And what they did is that they prevented this receptor from getting expressed. So those kids had a genetic disease, and we went and looked further, and we saw that the mutation they had were a little bit more frequent in East Asian, in Polynesian, and Tahitian as a heterozygote, meaning we have two copies. One is from mom and one is from dad. In their cases, the two copies were bad. And when we looked further, one copy was like the wrong copy in 2% of all of East Asian, Polynesian, and Haitian. There's a founder chromosome. And actually, probably this mutation arose, like sickle cell or other things, as a way to help us fight against infections. So this is why it got selected. And in their case scenario, it's leading to a disease that's called a lymphoma. So from one family, we identified other patients, and we have now more than 50 families and 50 different patients with this, and there's more to come. But more to that, we unleashed a place within this molecule that could be used for immune therapy, for patients that have cancer, that are dying, and would need something to even further unleash their immune system. We're pointing the way to a place in this protein that could be targeted and that would help unleash their immune system. So the other message is, by really being curious, by trying to understand and dig deeper from one family and two kids, you help those kids and others that are like that, and you understand the function of a very key protein that you could use to manipulate and help in cancers, in autoimmune disorders, potentially in the exhaustion of T cells in HIV and in MS. So again, being curious is sometimes helpful. And I hope this will create a little bit of some vocation to become scientists and doctors. And this is something that I think is, as long as you're passionate for something, this is what you need to do. And I'm here to ask any questions, to answer any question.
Okay, thank you very much. We do have time for a few questions, not too much, unfortunately, but certainly go ahead. We have the microphones.
Here, there's a question. I'll repeat it if you could say it without the...
You mentioned MS, and I was wondering, like, there's still no cure for it. Do you think we might find a cure for it in the future?
Okay, so the question is, you mentioned MS, there's no cure for it. Do you think there will be a cure for it in the future? I have absolutely no doubt, yes. There, even now, there is a new therapy that's oncoming. You know, there are things, MS is not only one disease, it's probably multiple different diseases within one, and some will answer to things and other will answer to others. There's enough research and enough efforts that already have prolonged and improved the quality of life of patients with MS. And we're very close to a cure because we're understanding even better the immune system. I wish the cure was today, like for cancer, but it's happening. The progress that was made in the last 10 years is phenomenal.
Another, anybody else?
Here?
What sorts of treatments have you developed so far to, manipulate the epigenome, as you were talking about, to treat certain diseases like brain tumor?
Okay, so what sort of treatment have been designed to manipulate the epigenome in those diseases? There are a lot of, so again, what we found is a mutation in the histone on a mark that gets modified. So what we have done, for those modifications, you need an enzyme to put a metal mark or to remove it. So there are drugs, and pharma have developed drugs to inhibit those enzymes because we cannot act yet on this. More to that, we are partnering with a new company, and this is why anything that you do in science is even, it doesn't have to do with drug development, that are using small molecule to degrade specifically the mutation. So this is where we're going at, to really selectively target the mutation or what mediate the post-translational modification.
Selectively targeted in the DNA?
Yes. And a lot of, so for example, this company developed a treatment that's currently it's FDA approved just recently for Creutzfeldt-Jakob, which is, as you know, the prion disorder.
Okay, maybe one more question, if we have one.
Here.
For stomach and brain cancer, total removal and subtotal remover, like I was told a subtotal remover, does it grow back, or total remover, like does it like ends there without?
So there are a lot of, brain cancer is not one. And actually in kids, there are as many brain cancer as there are different cell types within our brain, and this is where we have the most amount of cell types, actually. There's probably more than 300 different types of brain cancer. So for some of them, the low-grade ones, just removing the tumor is more than sufficient. It will never come back if you could remove it completely. But for the higher grade, you need more. Have I answered your question?
Okay, so I think we should thank both of our speakers, Dr. Jabado and Dr. Kay. And certainly for a very stimulating morning, and I think not to put too fine a point on it, food for thought. And I hope that all of you remember some of these things and think about them, talk to your peers, talk to others, and consider your future. You can do it, and you can do what you want to do with a passion that you've heard described this morning. So finally, Kate Duncan will talk to you briefly about programs for coming up. I'm not sure if it's the rest of the day or just for a short time, but she will give you your marching orders. And thank you again for coming. We really appreciate your involvement with these programs. Thank you.